Okay, here we go. Punnett squares. So we're going to use probabilities to explain the variation and distribution of expressed traits in a population. So traits are things that can be predicted based on the parent trait. Okay, so based on the two parents of an offspring, we can predict what their traits might be. So when the genotypes of two parents are known, it is possible to predict the genotypes and resulting phenotypes of the offspring. Genotype is the combination of two alleles for the trait, or for each trait, that each parent has, or each person has, or each organism has. The phenotype is the actual trait that is expressed, that we can observe in that organism or in that person. Okay, so there are differences between phenotype and genotype, right? Okay, so phenotype is what is expressed, what we can observe, what we can see, but the genotype is what does it actually look like at the level of DNA on that chromosome, right, in those alleles. Okay, so the different expressions for these characteristics are called traits, and traits are determined by alleles. The combination of the alleles from the parents becomes the offspring's genotype and determines what traits the organism will have. All right, Punnett squares, monks, and peas, oh my. So it was Gregor Mendel who observed traits in pea plants and did experiments that is really the basis for modern genetics today. So the Punnett square was named after Professor Reginald Punnett from 1875. He died 1967. It's one way to find and analyze the possible gene combinations of offspring using probability and statistics. Punnett squares were created through the work of Gregor Mendel. His studies of pea plants laid the groundwork for modern genetics. So he was looking at traits, okay, the color of the pea plant, the shape of the pea pod, like if it was wrinkled or smooth, 7 traits in all. Okay, he looked at. So he conducted breeding experiments of pea plants and concluded that some characteristics such as height, flower color, or pea shape were determined by specific factors. Law of dominance. So in Mendelian genetics, letters represent the genotypes of the individuals. In other words, the letters show which alleles each individual carries. So one letter stands for one allele. The phenotype, on the other hand, is the physical expression of the alleles and is the outward appearance of the genotype. So, law of dominance, okay? A capital letter means an allele is dominant. So these two parent 1 and parent 2, are what we call heterozygous parents, or heterozygous alleles, because they have the dominant and the recessive trait or allele for this trait. So a lowercase letter means an allele is recessive, and a dominant trait will always be expressed over a recessive trait. This is known as the law of dominance. One of the characteristics studied in Mendel's breeding experiments was the texture of the peas. Smooth textured peas represent the capital T and were dominant. Wrinkled peas represented by the lowercase letter t were recessive. Okay, so if I have 2 heterozygous parents here, they would both be, because of law of dominance, expressing their phenotype would be the dominant allele here, capital T, which would be, they both have smooth textured peas. Now their offspring, because they are carriers of that recessive allele, it would be possible for their offspring, only 1 in 4 or 25% of them would have that phenotype, the expression of the recessive allele. which would be the wrinkled peas. Okay, so it's 1 in 4 of these. Okay, all the other ones have a dominant allele. So law of dominance tells us that that would be what would become expressed. Okay, so here in the figure, we're showing a monohybrid cross. It's a cross for a single allele. It's represented by uppercase letter T is our dominant allele, right? Lower T is recessive allele. In this monohybrid cross, the parents are both heterozygous, like we talked about. So each parent has 2 different forms of an allele for that trait. Their genotypes are written across the top and down the side, and then we combine them. The genotypes inside the squares represent possible allele combinations for the offspring. So our genotypic ratio tells us the allele combination of the individual. It lets us know what genotypes are produced between the 2 parents. The phenotypic ratio shows us which traits are expressed based on the genotype, and these ratios are often different. Okay. We can also do a dihybrid cross. This is the possible inheritance of more than one trait, such as red hair and freckles. Okay, so here we have multiple alleles that we are crossing, right, in our Punnett square. Okay, so here we have a parent that is homozygous for recessive and then homozygous for the other trait, right? So there's 2 traits here. One parent is, if they're the same, like RR would be homozygous, capital F would be also homozygous. Okay, so then these would be the possibilities when we cross those for having red hair and freckles. So probability of traits is about the gene pool of a population being made up of all the alleles at all the gene loci, like location, in every single member of that population. So for example, our horses here have different colors represented by the gene pool of those particular horses. This means that every allele in the population has a frequency or a specific proportion. The alleles found in a population generally have several possibilities. So we're going to go through an example of finding the probability of an allele in a population. Given a crop of 500 pea plants, 280 have purple flowers, 130 have white flowers, and 90 have pink flowers. So we can find the probability of alleles in a population using an equation. Okay, so P would be one version of the allele, Q is the other version of the allele, and then P would equal the number of that type of allele divided by the total number of alleles in the population, and then we'd use this equation and subtract 1 from P. Okay, so we're going to assume that P equals CP, or purple flower color allele, and Q is CW, white flower color allele. What are the allele frequencies for the purple and white alleles in that population? We know the following. Plants are diploid, 2n, so the total number of alleles in 500 plants is 1,000, 5 by 2. All purple flowers have the genotype CPCP. All white flowers have the genotype CWCW. And let's see if my menu will go away. Hang on. All of the pink flowers have the genotype CPCW, so they're a combination. Okay. So we're going to start by finding the frequency of P. So P equals CP, purple flower color allele. We're going to determine what colors have a CP allele. Purple flower plants have the genotype CPCP, therefore 2 CP alleles. Pink flower plants have the genotype CPCW, therefore 1 CP allele. Take the number of organisms in each group and multiply by the number of alleles in that particular genotype. So 280 purple flower plants times 2 because of our pink flower plants. And then 90 times 1 is going to equal 90. Okay, so the total of the CP alleles is calculated by adding the number of alleles found in each group of flowers. So 560 plus 90 is 650 CP alleles, purple color flower allele in this population. To find the frequency of this allele, we divide the number of purple flower color alleles by the total number of alleles in the population. So 650 divided by 1,000, we said we had 500 plants. Multiply that by 2 because they're diploid, we'd have 1,000 alleles. So we get 0.65. Multiply by 100 to get a percentage. So 65% of that population have this CP allele. Okay, so now to find the frequency of the white flower color allele, we are going to use the equation to find Q. So Q equals 1 minus P. Q is going to equal 1 minus 0.65, so 0.35. We're going to multiply by 100 to get a percentage, 35% of the population. So whether we're working with 2 alleles or more, the frequency of alleles must always equal 1. 35 plus 65 equals 100, or 0.65 plus 0.35 is going to equal 100. So percentage of alleles in a population must always equal 100. Our frequency of P flower color allele, we add these two, right, is equal to 1, or percentage is 100. All right, quick summary. Traits are passed from parents to offspring. Using Punnett scores, we can calculate the probability of a certain phenotype, like a ratio, 1 in 4, right, for recessive phenotypes. And so we can calculate the probability of a certain phenotype, the expressed trait, using genotypes, the 2 alleles for a trait from the parent. A population will have an allele pool of possibilities of traits that can be passed on. Okay, see you on the next video.